Hello everybody. Today I'll be talking about the OTF lab and what we'll be doing for this experiment and then what will follow in the weeks past. So for this experiment, you will perform it individually, then you'll collect your own data, then put it into a Google Docs as a class. You'll analyze that data and then you'll be in groups of 4 and write not a lab report, but a scientific article on this procedure and your results. So you'll be in a group of 4 or 3 depending on the lab size you're in. And then for the general procedure, you'll be creating artificial food patches on index cards. The food patches will be soaked cotton balls in sugar solutions. You'll count and observe the number of ants that recruit to each food patch over time. After 1 hour, you'll count the number of ants on each patch, and then note whether or not there are more than just fire ants present and the relative number of other ants or insects at each patches. So you can identify any bugs or organisms by visual characteristics. You don't have to say their species. So if it's like a small black ant, you just say small black ant. If it looks like a fly, just say fly, something like that. At the end, you'll put on gloves and pick up your cotton balls, index cards, and put them in a bag. So, experimental design. A good experimental design in a manipulative experiment will include changes that only affect the variables of interest. You'll manipulate each variable independently. You'll exclude any other sources of variation as possible, and you'll have a control. So for our specific experiment, we're going to manipulate sugar concentrations of our treatment. So you'll be given 3 index cards, and then 3 cotton balls, one of 0%, which is our control, one of 5% sugar solution, and then one of 20% sugar solution. You'll be doing a replicate of this as well, so you'll have in total 6 cotton balls and 6 index cards. Then at the end, we'll combine all of the data for the entire lab and into one Google Docs, and then we'll analyze it independently. This week, we'll be doing the experimental part of this, so we'll be going out into South Campus. And then after Christmas break, we'll be doing the data and analyzing part of this and the essay writing part of this. So variation in field experiments. Where does it come from? So you have natural field variation, which can be inherent spatial variation in a natural environment. So where there are patches or homogeneous foraging patches. You have temporal variation, so weather or morning and evening times. And you have animal behavior. Experimental variation can be data collecting error or biases. You have random events that can mess up your experiment. And you have poor experimental design. A properly designed experiment will reduce the potential effect of other variables by trying to control the variation inherent in the environment, and you'll attempt to reduce the sampling bias and variation inherent to experimentation. So, manipulative experiments modify, add, remove individual variables of treatment in response to isolating the cause. So it's not just correlation. So you're going to have a manipulated variable. You're going to have a control or unmanipulated variable. You're going to have replicates, so repeated experimental units of each treatment and control. In this lab, our replicates will be the number of patches placed out per treatment. So it's not just 2 because you're performing 2, but it's the amount the entire class performs. So however many people in the class times 2. The sample size is the total number of patches you create. So 3 times the total number of replicates. So how many treatments are there? This is an example. So you have treatment 1, treatment 2, and treatment 3. Then what is the number of replicates per treatment? So you have 4 replicates per treatment. Then the overall sample size is the overall number of plants in this, so 3 times 4. Types of controls. In this experiment, you should have one or both of the following. So you have true controls, which usually leaves all variables unchanged. Then you have procedural or negative controls, which mimic some aspect of manipulation, which may also affect the outcome. Accounting for variation on controls. So sometimes manipulating organisms themselves can cause an unintended effect, which we'll call experimental artifacts. So disturbing an animal while they're moving or caging. Then experimental procedural controls, additional control experiments that examine the effects of artifacts. So for instance, cages are meant to exclude organisms that can cause shading effects on plants. Treatments. To ensure that your variable of interest is the only variable manipulated, you should ensure that your treatment and the control experience similar conditions for all the other experimental and experimental aspects. This is accomplished by spacing and randomizing your treatments. So spatial interprecision. So the hypothesis is the presence of owls will reduce squirrel foraging. So you see here presence of owls, and then you see squirrel foraging in the blue squares. So you would like to randomize it throughout the environment so it's not just grouped along the tree line and it's not just grouped out in the field. You can achieve interspeciation by randomly deciding where treatments are placed. So take a coin to your side, flip your heads or tails, or do a random generator. Take home point, make sure that each member of your group is not always placing the same treatment in the same location around an ant bed. So to avoid confounding variables, whenever we perform this experiment, do not just place certain treatments in certain areas that are different from each other. So, for instance, you see in this image here that there's a trail going through the forest, and then there's forested patches with leaf litter all around it. If you were performing this experiment, you would not just like to place certain patches on the trail and certain patches in the forest. You'd like to either have them all in the forest or all on the trail. So site selection, keep as many variables the same as possible other than that what you're manipulating. So pay close attention to variables at the site, so footpaths, trees, that might affect your experiment. You're going to write down your observations, so you're going to write down your GPS coordinates, you're going to write down site information, and you're going to write down ant observations. This is an example of what you would write down, and this is very important, especially for this quarter, because you're going to do this experiment and then go on Christmas break, so you're going to need as many notes as possible. So data to record during experiment. 1, the number of ants at each patch after 1 hour, and the 15-minute, 30-minute, and 45-minute time intervals. You're going to take a time of place patchment placement and patch pickup, the number and species of ants observed in the patch range during observation, the GPS location of every patch, then other metrics that will be hypothesis dependent but may include food type, tree girth, human activity in the patch range, and other relevant variables. So all data collected goes into optional foraging field results worksheet. So each person should have a worksheet to collect their data, then each person should have at least one patch for each treatment to monitor.